Hi-C is a method of identifying long-range chromatin interactions in an unbiased genome-wide fashion. First, cells are fixed with formaldehyde. Cells are lysed, and the DNA is subsequently fragmented with a restriction enzyme. Next, a biotin-elated residue is incorporated as the 5' overhangs are filled in. Blunt-end ligation is performed under dilute conditions that favour ligation events between cross-linked DNA fragments. The library is sheared, and the junctions are pulled down with streptavidin beads. The purified junctions can subsequently be analysed using a high-throughput sequencer, resulting in a catalogue of interacting fragments. These results provide us with insights into the folding of chromatin, revealing the presence of chromosome territories, the compartmentalization of the genome in open and closed chromatin, and evidence for the fractal globule organization at the megabase scale.
Hi, I'm Nienke van Berkem from the laboratory of Joop Dekker in the program of Gene Function and Expression at the University of Massachusetts Medical School.
Hi, I'm Erez Lieberman-Aden of the laboratory of Eric Lander at the Broad Institute of Harvard and the Massachusetts Institute of Technology.
I'm Louise Williams from the Molecular Biology R&D group at the Broad Institute.
And I'm Maxim Makayev from the laboratory of Leonid Mirny from the Massachusetts Institute of Technology.
Today we will show you a procedure for genome-wide chromosome conformation capture called Hi-C.
We use this procedure in our laboratory to experimentally explore the architecture of the human genome.
The first part of the procedure we'll show you will take place at UMass Medical School.
And the second part will be done at the Broad Institute.
We'll also show you how we've used the results of the Hi-C experiment to simulate the dynamics of the genome at MIT.
So let's get started.
Hi-C begins with cross-linking of cells, which is a common method in chromosome conformation capture. To begin, grow between 2 × 10⁷ and 2.5 × 10⁷ mammalian cells, either adherent or in suspension, and cross-link the cells. Lyse the cells in 550 microliters of lysis buffer using a homogenizer. Spin the chromatin at 5000 rpm and wash the pellet twice with 500 microliters of NEB buffer 2. Aliquot into 5 numbered tubes and add NEB buffer 2 to a final volume of 362 microliters. Add 38 microliters of 1% STS, mix carefully, and incubate at 65 degrees Celsius for 10 minutes. Place tubes back on ice immediately and quench the SDS by adding 44 microliters of Triton X100. Mix carefully. Add 400 units of Hindi 3 and incubate at 37 degrees Celsius overnight while rotating. The next steps are Hi-C specific and include marking the DNA ends with biotin and performing blunt-end ligation of cross-linked fragments. This step will allow ligation junctions to be purified later. Tube 1 should not undergo the biotinylation step and should instead serve as a 3C control to ensure that digestion and ligation conditions were optimal. To fill in the restriction fragment overhangs and mark the DNA ends with biotin in the remaining 4 tubes, add DATP, DGTP and DTTP to tubes 2 to 5. Also add biotin-elated DCTP and Clenau. Mix carefully and incubate for 45 minutes at 37 degrees Celsius. Following incubation, place the tubes on ice. Then, add 86 microliters of 10% SDS to all tubes to inactivate the enzymes. Incubate the tubes at 65 degrees Celsius for exactly 30 minutes and place them on ice immediately afterwards. To favour ligation events between cross-linked fragments, the ligation is performed under extremely dilute conditions. Working on ice, add 7.61 milliliters of ligation mix to each of 5 15 milliliter tubes. Then, transfer each digested chromatin mixture to the corresponding tube. Add 10 microliters of T4 DNA ligase to tube 1 to perform a regular 3C ligation. Then, add 50 microliters of T4 DNA ligase to tubes 2 to 5 to perform blunt-end high C ligation. Mix the tubes by inverting them and incubate for 4 hours at 16 degrees Celsius. Following incubation, degrade protein and reverse DNA cross-linking by adding 50 microliters of proteinase K per tube and incubating the tubes overnight at 65 degrees Celsius. Add an additional 50 microliters of proteinase K per tube the next day and continue to incubate at 65 degrees Celsius for another 2 hours. Cool the reaction mixtures and transfer them to 550 ml conical tubes. Purify the DNA in these tubes by performing a phenol extraction. Add 10 ml of phenol and vortex for 2 minutes. Spin the tubes for 10 minutes at 3,500 RPM and carefully transfer as much of the aqueous phase as possible to a new 50 ml tube. Repeat the extraction using phenol chloroform and precipitate the DNA using ethanol. After centrifugation of the ethanol precipitated DNA, each DNA pellet is dissolved in 450 microliters of 1x-TE buffer and transferred to a 1.7 ml centrifuge tube. Another round of purification is performed by doing 2 phenol chloroform extractions. Add 500 microliters of phenol chloroform and vortex for 1 minute. Centrifuge the tubes for 5 minutes at 14,000 RPM and transfer the aqueous phase to a new tube. After the second extraction, the DNA is ethanol precipitated. After spinning down the precipitated DNA, each DNA pellet is washed once with 70% ethanol and then resuspended in 25 microliters of 1x-TE buffer. Degrade any RNA that may be present by adding 1 microliter of RNase A per tube and incubating the tubes for 30 minutes at 37 degrees Celsius. Pull the high C contents of tubes 2 to 5, still keeping tube 1 separate as a 3C control. Now is a good time to examine the quantity and quality of the libraries by running an aliquot on a 0.8% agarose gel. Marking and ligation efficiency of high C should be evaluated with a PCR digest assay. The blunt end creation and ligation performed in the high C procedure will create restriction sites for the endonuclease NHE1. Amplified fragments from the 3C and high C libraries can be digested with Hindi3 and NHE1 to determine how well the blunt ends were generated and ligated. Some fragments will not have been ligated. Remove biotin from these unligated ends using the exonuclease activity of T4 DNA polymerase, as described in the written protocol. Purify the DNA again using phenol chloroform extraction and ethanol precipitation, and resuspend the DNA pellets in a total volume of 100 microliters of water. If the sample has passed the quality control assays, then the protocol continues with biotin pulldown and sequencing.
Thank you so much.
You are welcome.
To make the biotin-elated DNA suitable for high-throughput sequencing, the DNA must be sheared to a size of 300 to 500 base pairs with a Covaris S2 instrument. The sheared DNA ends are repaired by adding 10x ligation buffer, dNTPs, T4 DNA polymerase, T4 polynucleotide kinase, clinal DNA polymerase, and water. Incubate the samples for 30 minutes at room temperature. Following incubation, use a QIAGEN Minolute column to purify the DNA according to the manufacturer's recommendations. After loading the DNA on the column and washing the column, elute the DNA with 15 microliters of 1x TLE buffer twice. Then, attach DATP to the 3' ends of the end-repaired DNA by adding 10x NEB buffer 2, DATP, water, and exonuclease deficient clinal fragment. Incubate the reaction for 30 minutes at 37 degrees Celsius. To inactivate the clinal fragment, incubate the reactions for 20 minutes at 65 degrees Celsius and subsequently cool the reactions on ice. Using a speed vac, reduce the reaction volumes to 20 microliters. Next, load the DNA in a 1.5% agarose gel with 1x TAE and run for 3.5 hours at 80 to 90 volts. Excise DNA fragments between 300 and 500 base pairs and purify them with a QIAGEN gel extraction kit, using 2 to 4 columns depending on the weight of the gel. Combine the L08s from the QIAquick columns and bring the final volume up to 300 microliters with 1x TLE buffer. Finally, determine the DNA concentration with the Quantit assay using the qubit fluorometer and calculate the total amount of DNA. In this section of the protocol, ligation junctions are purified from the DNA pool, allowing for efficient identification of interacting chromatin fragments by paired end sequencing. Perform all subsequent steps in DNA low-bind tubes. Prepare beads for Biotin pulldown by washing 150 microliters of resuspended magnetic streptavidin beads twice with 400 microliters of 1x tween buffer. These and future washes consist of 5 steps: adding buffer to the beads, transferring the mixture to a new tube, rotating the sample for 3 minutes at room temperature, reclaiming the beads using a magnetic particle concentrator, and removing the supernatant. Resuspend the beads in 300 microliters of 2x no-tween buffer and combine with 300 microliters of high-C DNA. Allow the Biotin labelled high-C DNA to bind to the streptavidin beads by incubating the mixture at room temperature for 15 minutes with rotation. Reclaim the DNA-bound streptavidin beads with a magnetic particle concentrator and remove the supernatant. Wash the beads with 400 microliters of 1x no-tween buffer, followed by 100 microliters of 1x ligation buffer. Resuspend the beads in 50 microliters of 1x ligation buffer and transfer the mixture to a new tube. To prepare the DNA for Illumina paired end sequencing, take the total amount of DNA used as input for the Biotin pulldown, which was calculated earlier using the Quantit assay, and divide it by 20 to estimate the amount of high-C DNA that has been pulled down and is available for ligation. Add 6 picomoles of Illumina paired end adapters per microgram of high-C DNA available for ligation. Use 1,200 units of T4 DNA ligase to ligate the adapters to the DNA. Incubate for 2 hours at room temperature. Remove non-ligated paired end adapters by reclaiming the high-C DNA-bound beads and washing the beads twice with 400 microliters of 1x tween buffer. Wash the beads with 200 microliters of 1x no-tween buffer, followed by 200 microliters and then 50 microliters of 1x NEB buffer 2. After the last wash, resuspend the beads in 50 microliters of 1x NEB buffer 2 and transfer to a new tube. To determine the number of cycles necessary to generate enough PCR product for sequencing, set up 4 test PCR reactions with 6, 9, 12, or 15 cycles. Determine the optimal cycle number by running the PCR reactions on a 5% polyacrylamide gel and staining with cyber green, ensuring the absence of spurious bands and the presence of a smear between 400 to 600 base pairs, which is the length of the sheared products after ligation to the adapters. Amplify the rest of the high-C library-bound streptavidin beads in a large-scale PCR with the optimal number of PCR cycles. Pull the PCR products from the separate wells and reclaim the beads. Keep 1% of the large-scale PCR products separate to run on a gel. And purify the remainder of the PCR product with 1.8 times volume ampure beads according to the manufacturer's recommendations. Elute the DNA with 50 microliters of 1x TLE buffer and compare 1% of the ampure bead purified PCR product to the 1% aliquot of the original PCR product on a 5% polyacrylamide gel, ensuring the successful removal of the PCR primers. Sequence the high-C library with Illumina paired end sequencing. Align each end independently using MAC to identify interacting chromatin fragments. The following results are expected when the high-C protocol is executed technically well and can be considered quality control standards. Quality control steps should reveal that both 3C and high-C libraries run as rather tight bands larger than 10 kilobases. A DNA smear indicates poor ligation efficiency. Typically, ligation efficiency is slightly lower in a high-C library as compared to a 3C template. High-C marking and ligation efficiency can be estimated by digestion of a 3C PCR product. 3C junctions are cut by Hindi3 and not by NHE1. The reverse is true for high-C junctions. This PCR digest assay shows that 70% of high-C amplicons are cut by NHE1 and not by Hindi3, confirming efficient marking of ligation junctions. Analysis of the sequenced reads should show that reads from both intrachromosomal and interchromosomal interactions, indicated by the blue and red lines, align significantly closer to Hindi3 restriction sites as compared to randomly generated reads, shown in green. In a successful experiment, 55% of the alignable read pairs represent interchromosomal interactions. 15% represent intrachromosomal interactions between fragments less than 20 kilobases apart, and 30% are intrachromosomal read pairs that are more than 20 kilobases apart. This distribution may be sampled prior to high-throughput sequencing as a form of quality control. Cloning and Sanger sequencing around 100 clones is usually sufficient. The chromatin interactions can be visualized as a heat map, where the X and Y axes represent loci in genomic order, and each pixel represents the number of observed interactions between them. Typically, DNA fragments that are very close to each other in the linear genome will have the tendency to interact frequently with each other. This is seen in the intrachromosomal heat maps as a prominent diagonal. The following results show different ways of analyzing the data to reveal various levels of genome organization. Plotting the contact probability versus genomic distance shows that the probability of contact decreases as a function of genomic distance, eventually reaching a plateau. At every distance, intrachromosomal interactions, shown in the solid line, are enriched relative to interchromosomal interactions, represented by the dashed lines. This directly implies the presence of chromosome territories. Calculating the observed versus expected number of interchromosomal contacts between all pairs of chromosomes reveals preferential association between particular chromosome pairs. Small gene-rich chromosomes preferentially interact with each other, indicated by the bright red color. Individual chromosomes can also be examined. The raw heat map can be adjusted using an expected heat map to account for the genomic distance between pairs of loci, resulting in an observed over expected heat map. Then, a correlation matrix can be produced by correlating the rows and columns of the observed over expected heat maps. Using correlation analysis, it is demonstrated that the human genome segregates into two compartments. This is illustrated by the plaid pattern in the correlation heat maps. Using high-C data, new insights were gained into chromatin folding at the megabase scale. An ordinary polymer model of chromatin packing would suggest that chromatin packs into an equilibrium globule. Plotting contact probability as a function of distance illustrates that contact probability scales as a power law with genomic distance whose slope is approximately -1. This is not consistent with the behavior of an equilibrium globule, but does match expectations for an alternative structure known as a fractal globule. Here, two globular structures are shown. Coloration corresponds to distance from one endpoint, ranging from blue to cyan, green, yellow, orange, and red. Unlike equilibrium globules, fractal globules lack entanglements. In a fractal globule, loci that are nearby along the contour tend to be nearby in 3D, leading to the presence of monochromatic blocks. Such blocks are not found in the equilibrium globule. The lack of entanglements can also be demonstrated by removing the force constraining the globules and allowing the globules to expand. The fractal globule rapidly decondenses, whereas the equilibrium globule does not because it is too knotted.
We've just shown you how to make and analyze a high-C library. When doing this procedure, it's important to remember to do the proper controls to ensure efficient high-C ligation and purification.
The number of reads will ultimately determine the resolution of the interaction maps. We have used about 30 million reads to generate interaction maps of the human genome at a resolution of about 1 megabase. In order to increase the resolution by a factor of n, the number of reads needs to be increased by a factor of n squared.
We hope you've enjoyed learning about the structure of the human genome.
So that's it. Thanks for watching and good luck with your experiments.